All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. Today we're going to talk about a vaccine which some of the articles that I was looking through, they're calling it a super vaccine. And the reason for that is I think that they feel that this would take care of all coronaviruses. And I have gone through the technique and the methods and the design of the vaccine. And I think it is a very good vaccine, much better than the others. I don't think it is yet a super vaccine. So let us go through the vaccine article here. It's a very in-depth review of the vaccine. So let's break our discussion into three pieces. First part, I would discuss the summary of the vaccine test that they have done, researchers. Second part, I would go in depth of what the test was done and where does it work on SARS-CoV-2. And then the third part is I would talk about the design of the vaccine and the results of it. So you can decide to stay in for the whole session or go out after the summary or after the SARS-CoV-2 part or look at the design and method as well. So let's start. So here are gifts for humanity continue. And let's start with the vaccine itself. So here is a summary of the vaccine. So they are calling it a super vaccine. I think it is not yet a super vaccine. It's a pretty good vaccine, better than so many other that we are seeing. What are the core principles of this vaccine? The core principle is that can we develop an inexpensive vaccine? That is a core principle for all. Excuse me. Can this vaccine be produced in 2 to 3 weeks? So if you say them today that, hey, let's start making it, they have the platform and they have the countries where these kind of vaccines are already made, and these can be made within 2 to 3 weeks. Can we target somewhere on the coronavirus? So this is the coronavirus context, the SARS-CoV-2. Can we target somewhere on SARS-CoV-2 that the target on the SARS-CoV-2 is critical for SARS-CoV-2's function, and still we can target it, and it is not exactly just a spike protein. So that these are the basic principles that they used. The results that they did was that the vaccine was not sterilizing. It still allowed mild infection. It covers all coronaviruses, and that is beta coronaviruses and alpha coronaviruses and human coronaviruses and animal coronaviruses and various strains of them. This alone can be said that, hey, based on this, we should call it a super vaccine. And sure, then we would say this is a super corona vaccine, but still, I think there is still more work to be done. Then it uses a technology that has been already used for decades for pertussis and cholera. So it's not a new technology. Side effects are known, and building it is also known. Examples of such vaccines in the past, cholera, pertussis vaccines are already there. And there are factories for making these vaccines in many, many developing countries. And these factories can be adapted to start building, let's say, SARS-CoV-2 vaccines within 2-3 weeks. The cost is lesser than $1 per dose. So it is a very inexpensive and very rapidly makeable, if that is a word, vaccine. So this is the first part. This is the summary. Now we're going to look at how this vaccine works on SARS-CoV-2 and what is it that targets. So here we have a coronavirus. So what this vaccine does is it also targets the spike protein, but not the pieces that we already know. It is a new piece that we have usually not talked. I think we talked about it with bromhexin, but not a lot. So we'll talk about that now. And here the coronavirus is asking you to not watch my lectures. So is YouTube, so that you don't start killing the virus. So it is kind of saying, hey, don't do this. I think YouTube is also in the similar ballpark. Okay, spike protein. So let's say on that coronavirus above, we take this spike protein and look at its structure again from a high level. We know that the spike protein is a trimeric structure or 3-part structure. Our interest at this time is in these 2 parts, which are S1 and S2. The S1 part has receptor binding domain on it. That part allows the SARS-CoV-2 to bind with ACE2 receptor. And the S2 part has a protein part which we are interested in called the fusion protein. This protein allows the virus to fuse with our cell membrane. We have talked about it with the TMPRSS2 and bromhexin. We will briefly review that again. So keep the FP or fusion protein in your mind. Today's game is all about this fusion protein. We are not talking about the S1 part. We are not talking about the receptor binding domain. So here, if you see on the right side, let's say this is a coronavirus, and I have made these um, um, spike proteins with those divisions on it. We know that for a coronavirus to infect our cells, it has to have its spike protein primed. And primed means that the TMPRSS2 on our cell's surface will cut the S1 from S1 and S2 from each other, and they would both will become free. So then what will happen is once they are free, the S1 portion, if you see here, S1 portion will bind with this with the ACE2 receptor, and the S2 portion, this green leg, will fuse with our cell membrane, but this fusion will require this fusion protein part. So now think about it before we move forward. If for with some mechanism, if we can disable the fusion protein part, this red part over here that the virus is using to bind with our cell membrane, do you think then the virus can enter our cells? No, it cannot. It can bind with the ACE2, but it needs the help of the fusion protein to merge with our cell membrane. And if you block that part, then it cannot merge. So that is a basic principle. That is a core technique of this vaccine. Now, why is this a super vaccine? This is a super vaccine from this point of view that this part here, if I can circulate around, make a circle here, this part is pretty constant in all coronaviruses. Alpha coronaviruses or beta coronaviruses, animal coronaviruses or human coronaviruses, various strains of SARS-CoV-2, this part is 93% constant in all of them. That means if there are antibodies that are specifically generated against this part, not the rest of the spike protein, but this part, then even when the virus is present, it would find it very difficult to fuse with our cell membrane. Now, I know that some of the more medical folks would say sometimes it is not necessary that it fuses with the cell membrane. Virus can actually be phagocytosed. So let me quickly draw that as well. So let's see what we mean by this. So let's say here is a cell. It is a possibility that the SARS-CoV-2 is brought into the cell in the form of a vesicle, and it is sitting inside the vesicle. So it doesn't need to merge. It does not need to fuse with the cell membrane. We actually just pinch it in. So in this case, you could say, well, now this antibody is useless, but that is not true. For the virus to get out of this little vesicle, it still needs to bind with the membrane here and then come out. So that means the virus is going to need the fusion protein part to be functional and to be able to attach that to the membranes. And so if we can hold that hand, then the virus will not work correctly. So let's continue. So here, if you can see, the coronavirus is crying. Why is it crying now? Because this vaccine has generated antibodies against the fusion protein, and when the virus would now arrive in our body, those fusion protein antibodies will bind to all the fusion proteins, if you can see here as well, and the result is the fusion protein cannot fuse with the cell membrane, and then the virus cannot really enter the cell. So it is sitting outside, it is there, it is viable, but it is crying because it cannot enter a cell. And as you can see, the cell is pretty happy down here. Okay, so with this, now the second part of the talk is done as well. If you wanted to know what is the antibody about, this is what it is about. Now I'm going to go over the mechanism, how they made the vaccine and how they tested it. So here is the technique part of it. It's a pretty cute technique, and it is a pretty common technique as well. It's nothing new. So what they do is, we have talked about in our past uh, topics, we've talked about the recombinant vaccines or recombinant genetic materials. What that means is that you take a bacteria, and in that bacteria you insert the genetic material of your interest, and that bacteria will then make proteins that are made by that genetic material. That genetic material is inserted in the bacteria, and it is called plasmid. Plasmids are actually small DNA portions. We call them in genetics cassettes. So the folks, the researchers who work with them, they call them cassettes. Just like you have cassettes that you can put in a tape recorder and that cassette will be played. Similarly, these are little cassettes, and they have those, and they can insert them into the bacteria. Now, if a cassette is not present, then a new cassette can be formed. So here, first step to make the vaccine, get a bacteria that is modified. So in the case of this particular vaccine, they got an E. coli bacteria. Why did they get an E. coli bacteria? That bacteria is pretty harmless with certain modification. It can be pretty harmful as well, but it is pretty harmless with certain modification. So Tokyo Metropolitan University group has an E. coli that is modified. So modified means it can still survive, it grows very slowly, and it allows you to further modify it. It's not dead. Now, the things to do with the E. coli, this bacteria, we need to modify it to reduce the surface proteins on this bacteria. Just like, when we go for some surgical procedures and they would shave off the hair from our skin and make it clean. Just the same way, the researcher want bacteria that do not have its own proteins on the surface, that have less proteins on the surface, and then they can grow more proteins on the surface of which are of our interest. So less surface proteins, shaved bacteria, if you will, slow growing. So there was a bacteria there that was available. They took that and they further modified it to reduce the proteins even more on its surface. So what is the takeaway from this step? They had a bacteria and they shaved a part of it. Now, what do they want to do? On that clean part, they want to grow their own proteins on the surface of bacteria. This is just like, there are plants, you can take the branch from one plant and attach it to another plant, or we can do organ transplant from one person to another. Here, we want to add our own genetic material in this bacteria, and we want that genetic material and the result of that genetic material, proteins, to appear on a specific area on the bacteria. So that's what's happening. So what is that genetic material that we are going to add? So see here on this side, this little transporter. So all of these cells have channels in them. Channels are controlled mechanisms, doors, if you will, that allow things to come in or go out, but with certain conditions. Similarly, this E. coli has a channel called automatic transporter. The function of this channel is, imagine now, let's say you want to have a woman, for example, let's say they want to have a flower in their hair. So to put that flower over there, we can get a flower and just put it over there, but bacteria cannot just ask another bacteria to put a flower there. So what they do is they make their own tiny little flower or a protein, and then they, from inside of their body, it kind of emerges and grows on their surface, just like we have here. So this channel is used by the bacteria to bring on the surface of the bacteria the proteins of its own interest. All cells have various proteins on their surfaces, and these proteins do various functions for those cells. So when this bacteria will like to have a protein to grow on its surface, it uses this transporting channel to make the protein inside the cell and then bring it out on the surface. So the researchers, very clever, they are both from US, what they did was they created a modified transporter genetic material. They created the genetic material which will make the transporter, but this transporter, check this out, hear this out, this transporter would automatically have a protein attached to it, which is SARS-CoV-2 fusion protein. And when that modified plasmid, the modified genetic material, is injected in this bacteria would start making these channels, and these channels will then produce SARS-CoV-2 fusion protein, and these will be decorated on top of the bacterial surface. Why are they decorated on the bacterial surface? This is a way to present the antigen to our immune system. We are injecting bacteria that are decorated with the pattern that we want our immune system to attack. That pattern is the fusion protein. Now, could we use the same thing to do the rest of the spike protein? Yes. These researchers decided that fusion protein is the commonest, most constant part of the spike protein in all of the coronaviruses. So they just attacked that part. Okay, very good. So now we have a bacteria which has a channel that we are interested in. So we then go in, so researchers, and they create that cassette, that plasmid, we call it. Plasmid is that DNA material. That DNA material has been modified so that it can produce the protein of our interest. In this case, researchers created two types of plasmids, one that can produce SARS-CoV-2 fusion protein, and the second that can produce another coronavirus fusion protein, which causes diarrhea in pigs. So that was the Porcin epidemic diarrhea virus. So there is a Porcin epidemic diarrhea virus, which is also a coronavirus. It is an alpha coronavirus, not a beta coronavirus. And it's a pig coronavirus, and it causes diarrhea in the pigs. So what they did was they took the fusion protein from the spike protein of that alpha coronavirus and the spike protein from the beta coronavirus of SARS-CoV-2. Any strain doesn't matter, because all strains have them, or all variants or mutants have this as a constant. The reason they tested with these two is they wanted to see that, is it really true that if these are constant parts of the coronaviruses, then they would build antibodies that can then help each other. So that was their basic point, and so that's what they did. So here, this is the this is the bacteria. This bacteria has the cassette inserted in it. Bacteria, we have to make sure that after all those modifications, it stays alive. Plasmids usually do not kill the bacteria. Bacteria actually use plasmids as a superpower. For example, they have plasmids that can help them resist various drugs, antibiotics, and they can exchange those plasmids with each other as gifts. So bacteria are used to using plasmids. We are just using their mechanism to inject our own plasmid. Okay, so we injected the plasmid. Now this bacteria becomes the vaccine. And then we grow them. We cultivate them and we grow them, and then we bottle them up. And here we have a vaccine. Once we have bottled them up, so till this point, now we made the vaccine. So what is a vaccine? It's a bacteria on the surface of which there is SARS-CoV-2 fusion protein showing, decorated on it. And what is the purpose of that bacteria? When that bacteria will be injected in our body, it cannot cause disease because it is modified. It grows very slowly as well, so it cannot just be a foreign antigen growing. It would be killed by our immune system very soon. But in the meantime, the decoration on the surface will allow the immune system to become aware of the fusion protein part of the coronaviruses and make antibodies against it. So then any coronavirus that comes in should be attacked. I want to inject a seed of thought in your mind here. If this was really a super vaccine, then our own human coronaviruses at some point would have created the antibodies as well against fusion protein, and then that should have given us protection from SARS-CoV-2. Now, I am implying here that we don't have that protection, but who knows, there may be some people who do have protection because of fusion protein antibodies to human coronaviruses, which would protect against the animal coronaviruses too, including SARS-CoV-2. So maybe there are people who have it. So I just want to make sure that we keep it in balance, that yeah, it looks like super vaccine, but that may be a play on the headline as well to get your attention. It is a good vaccine, much better than others. I don't think it is yet super vaccine before we test it more. Okay, so number 6, how do we test it? So what they did was they took 21 piglets. These piglets were 5 weeks of age each. They divided them in 3 groups. And what they did was they injected this vaccine in their neck muscles. And they 3 groups of 7 each. One group of 7 received the bacteria that showed fusion protein for SARS-CoV-2. The other group of 7 received the bacteria that showed the fusion protein of the diarrhea virus, which is also coronavirus. And the third group of piglets received the dead bacteria without any decoration, just for a control to see what happens to them, so that they can compare the side effects. So that is what was given. So on day 0, the dose 1 was given, then a booster was given on day 21. A few weeks after, then they took the piglets. These were, I think, sacrificed. Their tissues were taken, their cells were taken, their blood and antibodies were taken. And here is they were challenged, before they were sacrificed, they were challenged with the antigen. So here is a coronavirus and it's saying, hey, what's up? I'm here. And then they saw that how did the piglet do. So in all cases, what they observed, all cases meaning that the one that were protected cases, not the control. In the protected cases, what they saw was that the animals did not develop severe disease. They still developed some symptoms, but clinically they were protected. They didn't become adverse. So that was done. However, what they saw was, and it is very interesting actually, it may not be bad, they were not able to sterilize the animal. Hey, Luffy. What happened? What? You sound angry. What happened? So he's happy that his anger has been acknowledged. So the the vaccine did not create a sterilizing capability, but it was still able to combat clinical disease, which is good enough. So not sterilizing, primarily T cell response. This is the other part, that they could not see B cell response, they could not see antibodies. They saw that cytotoxic T cells were more in number. So now how did the cytotoxic T cell, they can also bind with the fusion protein. All T cells, helper or cytotoxic or B cell or not T cell, B cells, they can all bind with the fusion proteins. So they saw that the T cell pathway was more active. And how did they find that? If you go here, this is the now famous diagram that we have probably memorized it by heart. So here is a vaccine. Vaccine goes into the innate arm system, and let's say this is a dendritic cell or macrophage that would present the vaccine, the antigens pieces to naive T cell. So here some decorations from the surface of the bacteria, which are fusion proteins, will be chopped up and presented to the naive T cell. The naive T cell can now go this pathway or this pathway. We have done this, T helper 2 or T helper 1. If it goes T helper 2 pathway in the presence of interleukin 4 and absence of interleukin 12, and then the T helper 2, the naive becomes T helper 2. T helper 2 then releases interleukin 4, 5, 6, 10, which then causes B cell to become activated into plasma cell, which will make the antibody. So that is one pathway. The researchers saw that this pathway was not taken predominantly. This pathway was taken. And how did they know it? They saw a lot of interferon gamma produced. Interferon gamma is produced by T helper 1, by cytotoxic T cells especially. It is produced by natural killer cells as well. But this is the primary area. So presence of interferon gamma tells a researcher that the pathway taken by the immune system is this pathway. So that's what they observed. So here, if we go back to the results, they saw that less B cells were active, more T cells were active, lots of interferon gamma were produced, significant protection from clinical disease was observed. And the important part here is you could give the fusion protein of any of these viruses, and it would provide cross protection from the other virus. That means if we have, let's say, wild type Wuhan virus, and then somebody gets SARS-CoV-2, let's say UK strain or India strain or South Africa or California or New York or whatever, they all have fusion protein which is similar in all of them. 93% similar, even across various categories of coronavirus. And because of that, there would be protection offered. So on one basis, I can call it a super protein, a super vaccine, and that is it protects from clinical disease from all coronaviruses. But it does not provide sterilization. It does not provide a lot of other things. It does have some disease that occurs as well. It does not have antibodies produced to the level that needed. And that may be a dose adjustment or maybe the decorating protein adjustment of the bacteria that may come in later on. So this is the discussion. I hope it makes sense. So let's do this. I'm going to stop here so that this lecture can stay short, and I'll come back in 5 minutes and we will talk about we'll do chit chat. So please do me a favor. Sterilization. WT sterilization means that the virus cannot be received or it cannot be shed by a person who is infected. This is not the sterilization of fertility. This is a sterilization of shedding the virus. And so they said virus was shed. It was in feces. It was present in the animal. So that is okay, actually, as long as the person does not get clinically sick. And then we can figure out what to do with the shedding. The vaccines that we have today, they also do not stop from shedding. They're not sterilizing. They are neutralizing, and the shedding still occurs. Shedding reduces, but not goes away. So with this, thank you very much for your time. Thank you for listening. Please like, subscribe, and share. There are 3 links in the description. One is for the, if you would like to buy me a coffee, that does not need PayPal. There is one more for being a patron, and one more if you wanted to support this channel. So thank you very much. And just a notice, if you're not going to be in the next talk, tomorrow in the morning, I will not be available because I'm doing a rehearsal with Dr. Teslari and her team for the conference. So thank you, and I would see you soon.